It is again my distinct pleasure to introduce the next speaker. We're going to be moving into the next segment of this session, is looking at how environmental factors or linkage to telomere sciences. And I really like to introduce my next speaker, which is Dr. David Gilley. He's the associate professor for in medical and molecular genetics and Indiana University, and he's going to talk to us about telomere dysfunction in breast differentiation, aging, and cancer, while you have the time to sort of get connected to the microphone. And again, it's a 25-minute talk and followed by a 5-minute question period. And we hope to keep the time so that we can have a long, integrated discussions after this session. So we have 2 more speakers.
Thank you. Thank you so much. It's very exciting to be here. I'm loving this workshop so far, and I thank the organizers for their insight into this area and bringing very interesting people together. And I'm looking forward to the talks after the talks, and hopefully we can spend some time doing that. What I'm going to talk about initially in the first part of my talk is really the consequence. So we've talked about, Maria and Alyssa have done a really good job setting up, so I don't have to give all those slides I usually give. But what's the consequence of telomere dysfunction? So you see shortening and all this stuff, and so what? What happens? What's the big deal? So there's been this concept, and actually Thea was one of the pioneers in her Nature paper several years ago, and Rhonda Penno, where you have a perfectly fine capped telomere, and some event happens where you get this uncapping event. Now, we've talked about shortening, but we can't really limit that. These telomeres are actually also sinks for oxidative damage, and there's other. We've done studies where the telomeres haven't shortened, but they become dysfunctional. And the hallmark of dysfunction of a telomere is when it fuses with another chromosome or it recombines in some way. Okay, so this is the readout. So you could theoretically have short telomeres and not see dysfunction. So it's more a little bit. We're sort of delving into this more now. So, and that obviously, if you get fusions of chromosomes, you're going to have a problem during the next cell cycle, right? You're going to lose a chromosome, you're going to gain a chromosome, you're going to aneuploidy, or you're going to go through these, as Thea talked about, the 1940s and 30s, Barbara McClintock talked about this, these breakage, fusion, bridge cycles. Okay, so this is really the consequence. So the question really a few years ago was, really, does this really happen? And what I'm going to focus on is really cancer today. And there was really a knowledge gap in the sense that there was no direct evidence of telomere dysfunction occurring in human cancers. There was a lot of evidence about shortening and the possibility of this happening. And to really answer this question, we had to, and others have talked about, develop a new tool to look at this. Because we couldn't just simply look at tissues and make metaphase spreads and drop the cells and see if we had fusions. But to look at actual human tissue, we had to develop a new method. And so this is the normal situation. We have chromosomes, and they're perfectly fine and segregating. But really, this is the marker that we wanted to look for, some linkage event. And I'm using linkage in a purposeful way. And so we developed a PCR-based assay, which really actually sounds trivial, and it wasn't that trivial because we're working in very repetitive regions. And so we're finally able to develop a method to do this and to amplify across these these fusion junction regions, and then look at detect fusions from human tissues. To do this, dealing with the human genome, there's a lot of complications, and so we developed sets of primers. And one of the problems with doing this is that not all the telomeric sequences are known. Almost half of the ends are not in any kind of genomic database that you can find because they're so repetitive. But there's quite a few that are known, and we focused in these TAR regions, which are a couple kilobases from the ends. They're right at the ends of chromosomes, pretty much. And currently we're covering about 40%. It's a multiplex PCR reaction. There are 1,080 different combinations of chromosomes that can recombine. So, and currently we're only able to cover about 15% of those possible combinations. But we're getting some interesting science out here, okay? And I'll talk about that. So we did a lot of studies. I don't have time to talk about that in cell lines that have known fusions. But what I'm going to talk about here is just that we see these fusions in breast cancer in an earlier stage in grade 3 right now, and also in invasive. And the levels of fusions, which is interesting, come up in this earlier stage, grade 3 DCIS, and then they're maintained throughout the more evasive stage. And about 40% or so of our tissues are positive at this point. But remember, I said that our assay is only covering about 15%, maybe 15 to 20%. Since some of the chromosomes ends are not known, we might be actually covering those because there's homology between some of those TAR regions at the ends of chromosomes. So if you extrapolate that, if you give me the gift of extrapolating that number up, I think this is an extremely frequent event. Once we get better and better, we get the information, we develop more primers. We're looking at something that's occurring very, very frequently. We've sequenced these junctions, and early stages of in the DCIS, we mostly see probably a non-homologous enjoining type mechanism because we don't see any microhomology. When we sequence the later stage, we see more microhomology, so there might be a different mechanism happening in the later, more invasive stages. And then I was talking to John Murnane at the break, and what we see is some of these insertions, and I'll talk about that. Especially in the more invasive stage, we see these very interesting insertions. And the sequences, the telomeric sequences that are fused are very short, a few hundred base pairs. But we don't know if that's indicative of what's actually happening at the fusion or if there's some processing going on at that point. And so what's interesting really about tumors, and it's kind of somewhat counterintuitive, and I want you to remember this because in the second part of my talk, when I talk about normal cells in the breast, you'll be reminded of this. So the hallmark of solid tumors that are telomerase dependent are that they have very short telomeres and very high telomerase activity. Remember, telomerase is the enzyme that's lengthening the telomeres, so it's a little bit counterintuitive. But that's the way it is in most tumors, most telomerase dependent tumors. So going back to these insertions, and what we've found, and we've done a lot of these, and we've used different methods, and some methods we see more of these insertions, are that there are these retrotransposon-like elements at the fusion of the junction between a chromosome and a chromosome. And there are probably these fairly complicated linkage events, maybe multiple breakage fusion bridge cycles, sort of reflected in the end product that we're looking at here. And often there are these line or sign elements and or these LTR retrotransposon-like elements. And they're very short, usually just a few hundred base pair chunk. And this is interesting because John found in Chinese hamster that if you create a dysfunctional telomere, these elements will link up at the dysfunctional telomere in Chinese hamster. And then Drosophila essentially uses this mechanism. They don't have telomerase. They use this type of mechanism to maintain their chromosome end. So it's interesting as far as the evolutionary relatedness or whatever, that we're actually seeing these things in spontaneous human tumors, cancers. We're not manipulating, we're just looking at the tumor. This is, so it's developed spontaneously in these tumors. So fusions are occurring early, and we actually see the hallmark of telomere dysfunction. And it's probably a very high prevalent genetic marker. And so I've talked to you about breast, but we're looking at prostate, ovarian. We also see fusions, and we see these in precancerous states too. So we think it's a very, very early switch. And it's persistent throughout, at least in breast right now that we know of, and also in prostate and ovarian. Comes up early, persists. It's a very neat marker. And also in the blood-borne CLL, there's evidence of fusions. The second part of my talk is about analyzing the telomere in subpopulations of the normal breast. So when you study breast in humans, you can get enough tissue from reduction mammoplasty to isolate rare populations of cells, progenitor cells, and different of epithelial cells. And so what we're finding, really, the take-home message is that there's a very interesting plasticity that's developed in the way in the telomere biology. There's shortening of the telomere, turning telomerase on, depending on the cell type, which is age independent, which I'll show you, and also there's an age dependency to this. So we wanted to really understand what was happening in the normal state in the breast before we jumped into tumors. So we'd have some baseline. And this is an interesting topic for those that are interested in the cell of origin of tumors, the cancer stem cell hypothesis. But let's look and see what happens in the normal state first before we jump into the tumors. Of course, we'd love to jump into the tumors, but let's back off and build a foundation here. And I think what I'm going to tell you about is probably uncovering in the normal breast. And if you look at normal tissue, what you're essentially seeing is disease-resistant pathways. And it might also be uncovering some vulnerabilities to normal tissues in certain subpopulations. So the hierarchy of the epithelial cells in the breast, which are the source of tumors cells, as Thea had talked about, are that you have the stem cells, bipotent progenitors, and then these myoepithelial progenitors, and then the mature myoepithelial cells. This is in the basal layer, and with the current markers that we have right now to separate and sort these cells, these all come out in one population. So there's really no, unfortunately, method right now as far as FACS or sorting or surface markers to just get this guy. When you get this population, this is probably less than 1% of the population in the basal cells that you're looking at. And then there's the luminal progenitor cells, which we can sort, and we can get an enriched, very pure population of those cells with sorting. And the mature luminal cells. Now, what's interesting about this is that there's been some really interesting studies, I think, especially the cell stem cell paper, which was on my reading list, where there's some good evidence that these cells actually might be the cell of origin for tumors. And there's a very nice study done on that with a stem cell from Smalley's group. So what we do in this experiment is we take breast tissue, and as quickly as possible, we try to sort those cells out with different markers, okay? And we can get 4 nice populations. This is work done with Connie Eves in the Terry Fox Laboratory, who's like the world's expert at this, and she's just amazing. It's a wonderful collaboration with her. And so we're trying to really look at as quickly as possible. We're not culturing the cells. We're taking them from the breast and then looking at them as quickly as we possibly can. And then we're looking at their telomere biology. So when we did that, when we had these isolated subpopulations, this was from a 20-year-old woman, we found right away that the telomeres start to fluctuate or change depending on the population. So there's something interesting going on here, and we're getting nice enrichment of these cells. And the cell line that really sort of decreases down to a relatively shorter telomere in normal development in the normal breast in a 20-year-old are these luminal progenitor cells. Remember, just keep in mind that it looks like those might be, from some evidence, the cell of origin of most tumors, okay? Just keep that in mind. There's a lot of controversy about that, I suppose, but there's some evidence that these are the cell of origin, not the stem cell. So there's this plasticity that's happening in the normal subpopulations of telomere length during normal development, even in a 20-year-old. And we looked at, I think about 15 different women at different ages, and using a couple different methods, the Southern blot, which is really the gold standard of telomere length, and then qPCR. And we found that the luminal progenitor cells really do shorten, and regardless of age, and I'll show you another example of that. And there's a very good correlation between our Southern and our PCR-based method for measuring telomere length. Now, one of the things, here's another Southern blot of telomere length. And one of the things that we noticed in our early studies was that if you just take total breast tissue, you really don't see these fluctuations, okay? Even if you enrich for the epithelial cells, because most of the cells are not the cell that you want to look at. But once you start to isolate these different subpopulations, you see this very interesting dynamics or plasticity in the length of the telomere. And one of the things that happens, and I'll show you this later, is that in older, somewhat older women, 46 is not old, that's young, but compared to 20-year-olds, okay? The 20-year-olds, the luminal progenitor cells crash down to very short levels. This is really very short, especially compared to model systems where we know telomere dysfunction can occur. We can induce telomere dysfunction in cells at that kind of level. But in the younger patients, the more mature luminal cells, increase in length back up here, but not the slightly older person in their 40s. And so here's sort of the typical scatter that you see. And these are different individuals with different genetic backgrounds, so you see some scatter. But with age, these mature luminal cells are decreasing. So this is actually an age, a change in the length of these mature luminal cells with age. And about 50% of this change is due to the telomere length. So we wanted to look at telomerase. And what we found was that telomerase is kicked on at very high levels in those luminal progenitor cells. Remember, I've said that they were very short in their telomeres, but it's very counterintuitive. But telomerase gets kicked on to close to levels of sort of our classic cell lines that we use to sort of compare telomere lengths. It's about 25% of HT1080 cells, but it's still really kicking on quite high. And we get these really nice co-localization of the protein component of telomerase with the telomere. And this is occurring in about 80% of these luminal progenitor cells. So it's not just a small little population. It's the majority of the cells. And what we found, too, when we measured telomerase was that there was this decrease in the activity of telomerase in the LP cells with age, which correlated nicely with the decrease in telomere length with the downstream differentiated luminal mature cells. So we had short telomeres in these luminal cells, and we thought, well, these are very short. What's going on? So we did a microarray analysis initially, and we found that there was quite a few differences in the gene expression, and a lot of DNA damage response proteins were coming up in the luminal progenitor cells that have these shortened telomeres. So you're getting a DNA damage response. And a lot of the telomere proteins were going down, probably because the shortening of the telomere. These were confirmed. One of the candidates that we were quite interested in initially was the MRN complex, MR11, RAD50, and NBS1. And because there's a lot of evidence, and we've worked quite a bit for many years on the connection between the DNA damage response and the telomere. So this got our interest. And so we went looking, and this was just confirmation, validation of our microarray results. And what we found was that in the luminal progenitor cells, there was a massive DNA damage response at the telomeres in these normal cells. But not the BC cells, not the basal layer cells, not the stromal cells, just the cells that have the short telomeres. We rip out of the tissue. We look at these things as quickly as we can. But there's this massive telomere specific DNA damage response. This is gamma H2AX, which is a classic marker for DNA damage, for the DNA damage response. And it's happening specifically at the telomeres. I don't have time to show you. We also see 53BP1, we see NBS1, MR11, and RAD50 also at the telomeres. And just to give you an idea of how frequent this is, if we look at, if we compare the luminal progenitor cells to the basal cells, there's a very large response, DNA damage response at the telomeres of these cells with an average of about 10 foci per cell. Almost, essentially all of the luminal progenitor cells have a telomere-specific DNA damage response, or they have TIFs, which is interesting because TIFs, telomere dysfunction-induced foci, have been around for a while, but we've only seen those by artificially inducing telomere dysfunction. But to see it in a normal tissue without manipulation is interesting and suggesting it might have a real biologic function. And it's really essentially all the cells in the luminal fraction that we get out from sorting from tissue. So what's the overall concept here? Let me try to put it together as best I can. What we think is happening here is essentially in the luminal progenitor cells, we have shortened telomeres, we have activation of telomerase, and then a telomere damage response at the telomere, telomere dysfunction-induced foci come up. And what we think this is probably mostly a tumor suppressive pathway. And of course, we're looking into this right now. A way of eliminating cells that have been around for a while, developed mutations, and similar to the concepts of Judy Campisi with cellular senescence. But it's a double-edged sword, potentially, where there's an escapee, a very rare escapee that has telomere dysfunction, genomic instability, and then goes into a full-blown tumor. So how does this relate to environment? So I was asked during one of the phone conversations. Let me just say that, again, the luminal progenitor cells also have, just like tumors, solid tumors, shortened telomeres and activated telomerase. And we're putting this together with the fact of evidence that the luminal progenitor cells may be the cell of origin of tumors from other labs. So let me just conclude in the sense that the disease consequences of telomere dysfunction, it's very clear right now that this is important for cancer. Cardiovascular, it's a little bit less clear in the sense that it might be an effect, it might be a correlation, but we've actually did some studies where we saw shortening of telomeres associated with a twin study that we did with hypertension and cardiovascular disease. It's a little bit less clear in my mind that the telomere is involved with cardiovascular disease, but it's very clear. There's a lot of evidence right now that it's involved with cancer. So let me sort of put this together in a way as far as the environmental factors and telomere dysfunction. So let me note again that telomere dysfunction can occur through shortening. It can maybe oxidative damage, epigenetic remodeling, and other things that we don't know about yet. And lump this into, excuse me if I'm using the terminology wrong here, but into more exogenous sources or outside of the cell or outside of the organism, like smoking, obesity that Alyssa talked about, socioeconomic group, et cetera, as opposed to fitting these into these boxes isn't that accurate, but just for discussion purposes, into more endogenous sources such as perceptions of stress, maybe metabolism, which I should have put in here, meditation, exercise, etc. And then what I've shown you in the second part is that this might also be involved with just cellular differentiation. That during normal cellular differentiation, it's mostly a tumor suppressive mechanism, likely, but that we do see telomere dysfunction in these normal LP cells, and that even during normal cellular development, as we tease out what's really happening in the complexity of tissue, we see this massive response of telomere dysfunction in these luminal progenitor cells. So let me just finish there. I want to thank Connie, who's one of those wonderful collaborations that you have where everything works smoothly and you communicate and it's gone really well. And her hotshot postdoc, Raj, and then Hiromi in my lab, Lee Ren, Satoshi, and Nazmal. Thanks a lot.
I'm sure we have a couple of minutes for some questions. Maybe we can wait for those questions at the joint session. Oh, there's one right coming. Oh, could you use the microphone, please? Yes. Oh, okay, I see. Good. I'm sorry. Yes.
People are shy.
Very nice talking. I just what I guess I'm struggling with is when there's no doubt about what you're seeing, and lots of people here are seeing, but to what extent are these telomere changes just reflecting a generalized a genomic instability, plasticity that is occurring? So similar to microsatellite instability that you could observe with mismatch repair mutations in non-polyposis cancer. You've got a generalized phenomena, and you've got a good assay for it.
What's the question again? Sorry, I understand your point.
I'm just saying this whole cause-effect thing, I'm having a hard time.
The cause-effect thing? Oh, that's always hard.
I'm more.
That's the ringer question that everybody.
Is this just generalized genomic instability is occurring as part of an aging process?
I didn't really have time to get into like all of the evidence about telomere dysfunction. I just showed you what's happening in human tissue, okay? But what some of the work I had to leave out just for time purposes is that you can specifically introduce telomere dysfunction in mice and you get cancers and things like that. So that sort of helps you a little bit, I hope, with the cause and effect relationship.
But you get cancer with microsatellite instability and non-polyposis cancer. But the microsatellite instability is a generalized secondary phenomena that's occurring because of mismatch repair mutation. Are you seeing the equivalent of microsatellite instability? And you have something you can measure. It's real, but it's not. It's really the genomic instability that's causing the problem.
Well, I'm not exactly sure your question because I see your point, and what really I think it's hard to I wouldn't get up here and say that we've proven the cause of genomic instability or something like that, but if you believe that in a genomic instability driver of cancers, then we can talk about it in the sense that we're showing an example of telomere dysfunction in tumors and early tumors, and the relationship that has, and that obviously is been shown to drive genomic instability. So you might have a subtler point here that I'm not quite getting it, but I don't think we're really far.
It's not driving genomic instability. It's reflecting genomic instability. Just like microsatellite instability is reflecting genomic instability.
Right. I think that's a good question. I'm not sure we can't definitively say it's the driver of genomic instability. Okay, that's a good point. Good qualification.
I think we can take that question further as for the general discussion session. So can we just take one quick question and quick answer?
So the quick one is, have you had the chance to look at normal adjacent to tumors to see if you see the same thing or if it's different?
Yeah, of course we've done, and I showed some of that. We've looked at adjacent tissue. We also we have the normal Komen tissue bank where we have thousands of normal tissues, and we haven't seen fusions. So that's the nice thing about it. A genetic change, like aberration like that, is powerful because we're not looking at ranges of molecules where it's hard to make a decision on the individual patient basis, individual sample basis. But so far we just don't see it. We don't see it in normal tissue. We don't see it in adjacent tissue. If you work with normal adjacent tissue, you always kind of wonder how normal that is too. So that's sort of another caveat. But with our prostate studies, it's really hard to get men to donate their normal prostate tissue. For some reason, I don't know why it is. And so one of the things that we have to use is adjacent, normal, histologically normal, as far as we know from the pathologist. And we don't see fusions in that so far with the numbers that we've looked at.
All right, so David, thank you so much. Now we understand the volunteer spirit of men and women are quite different.